This is Journal Club for the 19th of February 2026 at Science Academy at Holy Cross High School. Today we're doing a paper from Nature Communications, which was published last year. 3D imaging of human pancreas suggests islet size and endocrine composition influence their loss in type 1 diabetes. There were a lot of abbreviations and acronyms in this, so hopefully you guys will clarify that pretty soon now. But uh, Maggie and Sam, go ahead and take it away.
This is our paper. The title of our paper is 3D imaging of human pancreas suggest isolate I, islet size and endocrine composition influence their loss in type 1 diabetes, which Sam,
So basically, what the title of our paper is saying is that they took 3D scans of multiple human pancreases from people without type 1 diabetes, people with a single autoantibody for type 1 diabetes, and people with type 1 diabetes. And these scans suggested that the islet cells and what the endocrine system is made up of influence like how the cells will die, which lead to type 1.
okay. This is the anatomy of the liver and the pancreas. So, the The pancreas has like two main jobs of digesting food and controlling blood sugar. So, digesting food is mainly seen at the exocrine portion of the pancreas down below. So, you can see it labeled with duct and acinar cells, which make the digestive enzymes. And then in the endocrine portion over to like the right, that is what helps control blood sugar. So, you can also see the islets of Langerhans, which are clusters of cells that play like an important part in diabetes. So the next slide kind of goes into it a little bit more in detail. So you can see that the endocrine cells of the islet of Langerhans, it consists of beta cells, alpha cells, and delta cells. So the beta cells produce the insulin, and the alpha cells produce glucagon, and the delta cells produce somatostatin, right?
Somatostatin.
Okay. And yeah, so the insulin and glucagon are most important in regulating fuel metabolism.
This is a diagram of the islet of Langerhans.
Hey,
And
Sam.
yes,
Sorry, before you get into that, can you go back a slide? No, back. Back.
For some reason, it's not letting me go for the back. So give me like 2 seconds.
Just you can use your arrows. The arrow
Um,
keys. Use the left arrow key.
there
It'll
we
go
go.
back.
Okay.
Okay. So, go back again. I want to see the pancreas. Just a quick clarification. Maggie said exocrine versus endocrine. Exocrine is when any organ or cell releases a substance that does a thing. Okay. So, those duct cells, for example, will release sodium bicarbonate, which is baking soda, just a solution of baking soda. And notice where it goes. I pulled out my tablet, so I should be able to do this. Hold on, I want to show you. Because the basics, in order to understand why this is cool, you kind of have to understand these basics. So, right here, it's not drawing. Come on, you could do it. Right? Okay. It's ignoring me. Let me try it this way. Ah, can you see where I just drew that? Y'all, yeah? Okay. It's a really bad color. Pick, say, red. There we go. Okay. So this duct here runs into the duodenum or duodenum. I pronounce it duodenum, which is the beginning of the small intestine. And notice that the small intestine starts right after, right here, where the stomach meets it. But the stomach's super, super acidic, right? So it's about pH 2. And then it goes and it dumps all of that chyme into the small intestine, which would damage it really bad if there wasn't some sort of basic substance to neutralize it. So these duct cells here produce an exocrine signal, which is sodium bicarbonate, which then goes and neutralizes the acid. So that's an example of an exocrine function. Whereas an endocrine function is what they're about to talk about, where you release a hormone that has then an effect on something else. So it's like, yeah, you're an organ, you're releasing a substance, and then that substance is going to have an effect on a different organ or tissue. That's what endocrine means. And again, just to be more specific, this thing right here is what you're about to see with Sam. So I just wanted to make that clear. Okay, go ahead, Sam.
So this is a diagram of the islet of Langerhans. And so what it's saying is in two different instances, when your blood glucose is high and when your blood glucose is low. So the purple cells are the beta cells, the ones that produce the insulin. And the green cells on the outer edge are alpha cells, which produce glucagon. And insulin is going to lower the blood glucose while glucagon is going to higher it. So when your blood glucose is high, those alpha cells are going to slow the production of glucagon, while the beta cells increase their production of insulin, which lowers the glucose down to like the normal range where it should be. And when your blood glucose is low, the beta cells are going to slow their production of insulin, while the alpha cells increase their production of glucagon, which higher your blood sugar.
Okay, and before we move on, I was told that Sam has a prop to share with the class.
I do have a prop. This is insulin.
Okay.
That's it.
So moving on. For figure one, who wants to
Hold on,
present?
wait. Sorry, I didn't realize mine was muted. Unshare your screen so we could see it full screen
Okay.
for the video, for the gram.
Mm.
This is insulin. It's basically just a clear liquid in a vial. It looks like water and it kind of smells like burning plastic. And that's
Now,
it for the
now, in order
demonstration.
now to get that insulin, do we go around like killing people and taking their pancreases?
Of course.
Okay, all right.
I think they, I think it might be from like pigs. I think that's when they first did it from.
That's what they first did it from was pigs. Yeah, no, nowadays they just grow it in E. coli. That's actually something that I thought about doing as part of the summer lab intensive was teach how to do that. If you guys were interested.
I think we
But
It
should.
sounds cool.
but instead, what you're about to talk about in the paper is what we are planning on doing next year, next summer in 27. Maybe not with the fancy equipment they have, but something similar to it. There
All
you go.
right. So do we have any volunteers for figure one, or am I gonna have to cold call? Okay, wonderful. Andrew, go ahead.
Okay, so we're looking at figure one, right? All right, so first we see these images right here. You get purple, blue, and white colors. What do they mean? So the purple, you see it says INS in the top left. That just means insulin. Insulin, obviously, we know it's a hormone in the pancreas, and it usually controls your blood level. But the blue stuff, GCG, is glucagon, which increases your blood level. So look at the top here, you'll see there's three groups, ND, GADA, and T1D. ND, I'm pretty sure that means just healthy, normal pancreas. And then the purple right here, GADA, pretty sure that means at risk. Obviously, still pretty healthy, but still at risk. And then T1D stands for type 1 diabetes. In ND, you can see that the insulin is pretty high. There's a lot of insulin in the pancreas. In the purple group, with risk, there's slightly less, but there's still a good amount. In type 1 diabetes, however, there is barely any insulin. There's like, maybe 5 dots of insulin that you can see there. Now moving on to the glucagon. In the normal healthy pancreas, there isn't much compared, to the type 1 diabetes. There's bigger spots in type 1 diabetes. There's also a lot in every single row, meaning that there's a much higher ratio of glucagon to insulin in a person with type 1 diabetes than someone who's at risk or has a normal healthy pancreas. You'll also see there's INS and GCG both in one group. That basically means it's an image and a scan of INS and GCG in one. So again, you'll see that there is more insulin in the healthy livers than type 1 diabetes. Now in the third row of each group, you'll see there's also white dots. And there's abbreviations for them, INS plus and GCG minus. I think that says INS plus and GCG plus, and then INS minus and GCG plus. Basically, this is just, short for higher to lower. So in the INS plus GCG minus, that means it's showing, more like how much insulin there is compared to glucagon. In the INS plus and GCG plus, it's saying just equal ratio of insulin to glucagon. And then, obviously, I'm sure we can figure out that INS minus and GCG plus is lower levels of insulin compared to higher levels of glucagon. So in the healthy pancreas, we see that there's there in the equal amounts of INS and GCG, it's there's a lot of dots in both the purple group and obviously the healthy group. However, again, in type 1 diabetes, there isn't as many dots. Now, moving on to the graphs below these images, starting off with 1BB, is just the amount of insulin.
Hey, Andrew,
Yeah.
just one second. I just want to make sure everybody's. I got confused by all of the abbreviations and the acronyms and stuff. Just ND means no disease or no diabetes, right? Sam and Maggie, right? GATA means what?
At risk for diabetes.
And was that the stage 0 one? Did it say stage 0? There's a new staging thing that they do for diabetes. And I kind of want to know, and this, Andrew, you're probably not going to be able to answer this,
Yeah.
but I kind of want to know what the purpose of this figure was. Like, why? Why show it to us?
So they can like see like how the cells are working and how much of what they're producing is.
But why? Why would you need to see that? Kira?
That's not really what I was going to answer, but
Oh,
GADs are what it means is glutamic acid decarbase antibody.
decop. Yeah.
I think I
So,
just messed up the name, but yeah.
yeah, that's glutamate decarboxylase, which is an enzyme involved in turning glutamate into uh, glycogen, I believe. So, and the GCG is short for glycogen. I don't know why. The INS uh, is not.
I thought it was glucagon.
Well, what did I say?
You said it was glycogen.
Oh, sorry. It is glucagon. I misspoke. It is glucagon, not glycogen. I don't know why I said that. And before I think I might have said glycogen also. I meant glucagon. Glucagon's just the other hormone. Okay. And if any of you are struggling for a reason, right, at this point to say, why do I care about this at all? If you or you know anybody who's on Ozempic, the semaglutide or any of those other drugs, they directly affect these two hormones. So, so INS is insulin. GCG is glucagon, and then T1D is type 1 diabetes, which I've never heard
Yeah.
it called that. Right. Okay. But I do want to know. So, Andrew, you can continue, but I want somebody to come up with a reason for why they did figure one. Why do we need to see this? Go ahead, Andrew.
Okay, any questions on the images right here before we move on? Any? All right, so going on to 1BB, the graphs. So it says INS, meaning insulin above. It's just showing how much insulin there is in each group. So no disease, high insulin. Um, the purple group, I'll just call it the purple group for now. There is pretty good amount of insulin in there, not too low, but in the type 1 diabetes, it drops down nearly to zero. Now in the glucagon, we see that no disease has the lowest, type 1 diabetes has the highest amount. So just telling us that in people with type 1 diabetes, there is a much higher amount of glucagon compared to insulin, which is why there's usually a much higher level of blood sugar. Now in DD, it's just showing the density. So it's basically telling us like how much of, you know, insulin and glucagon in the pancreas. Like how much is there in just one pancreas, right? So we see that in the no disease in the purple group, there is a much higher density of these molecules, all right, of insulin and glucagon. But in type 1 diabetes, there is not as much. So there are a lot less islets in type 1 diabetes. Islets, I searched it up, is just basically like all the hormones that are in the pancreas, which are just, for now, insulin and glucagon.
N-
Now,
An,
when,
an islet is a glucagon. It's a hormone producing
oh,
area, right?
okay.
They were the little, if you, Maggie, go back two slides. They're the, they're that thing, that whole image. Yeah, that whole round image. That's a single islet. And if you go back two or three more slides to see the actual there, so that those little purple spots that you see, okay, the whole group of spots, that's an islet. And there's lots of them. Okay, thanks.
Okay, so basically, there's less number of islets to produce these hormones, which is not very good in terms of density. Now, in E and FF, E is measuring how much like of these of the islets there are. So you see that in a healthy pancreas, 50% of islets, this counts for the knot disease and the purple group. You see that 50% of them usually have INS plus and GCG minus, meaning that there's more insulin than there's glucagon. You want that because you want your blood sugar to be controlled, and you don't want your blood sugar to always be high. Now, as you can see in the middle of the graph, INS plus, GCG plus, the ratios are pretty equal. There's when we're talking about the no disease and purple group, and it's still healthy. And then again, in the type 1 diabetes, it is pretty low. Now, in the INS, insulin minus, in the GCG, glucagon plus, meaning less insulin, high levels of glucagon, type 1 diabetes outranks them by a lot. Now, in figure 1FF, it is saying basically a pretty similar thing just in terms of volume, and you'll see the same results. Again, diabetes, much higher, glucagon, much lower insulin, and then for the rest, pretty even and normal.
So what's the point? This is for everybody, somebody.
The sugar can like affect your hormones.
Sugar. I don't know what sugar you're talking about.
The glycogen.
Oh, no, no, no. I made a mistake and called it glycogen. It's a glucagon. Glucagon's a hormone. That was my fault. Sorry, I was stupid and said the wrong thing. So wipe that from your brain, Asia, that I didn't mean to mention glycogen. Sorry. Dylan.
Dylan.
And hold on, Maggie and Sam, you guys should be facilitating. You're calling on people, right?
Dylan, go ahead.
I think they just wanted to uh prove that there was like a difference in composition. Not prove, suggest that there is a
Su-
difference in
support,
composition.
demonstrate,
What?
support,
Yeah,
demonstrate. Yeah, not, yes, I believe you're right. It's setting a baseline,
but
right?
I think we already know that. Like that's been known, right?
Right. Yes. But what's the impetus for this paper? Like, what's the point of the paper? I
They
mean,
wanted
even
to
in the
prove
title.
it was about.
Stop the word proof. We don't do proof. This is
Oh
not
yeah,
a legal setting.
they wanted to suggest that size had something to do with it. So
Size had something to do with it. Size of
it
what had
had
something
something
to
to do
do
with
with
I think he
of,
means the size of the
of this. Like,
yeah, the eyelets.
had something to do with diabetes.
that's the point of the paper?
Isn't that in the title?
It is. Wow. And I completely interpreted that title differently than you did. Um,
What did you think?
I also thought of it the way that Dylan thought of it, so
um,
Yeah, me too.
I think a point of it might be like to show which cells are like just removed by diabetes. Because obviously you see that insulin
right.
just goes somewhere.
Sure,
Diabetes removes them.
sure. But we knew that already. And that Dylan, like Dylan said, we knew that already. So why do it again in this paper? And Dylan, your answer was,
Well, I think they just wanted to use the 3D like modeling.
ah, yeah, I think so too. I think that's
They
the
wanted to
actual
prove that
thing.
it works.
They wanted they
Not prove,
wanted to,
suggest.
well, they wanted to show that it was better than the previous stuff. I think what Dylan said's right. I think that they had a piece of equipment that they thought would actually do a better job. And then the rest of this paper is really about showing it off. Fair? Maggie and Sam?
fair.
Okay. All right.
All right, anyone figure two? Kira, go ahead.
You're
No. Okay. So what figure two is talking about? So the general basis of it is its question is do gut hormone cells change during type 1 diabetes or if you have it? And a short answer to that is yes, they change a lot. So as Andrew said earlier, they use the same kind of like what are they called? Like abbreviations or like
Yeah, this one irked
acronyms.
me. This one bothered me a little. Yeah.
Okay. Do you want me to go over them again or do you think we're
Yeah,
good?
because yeah, I did not like EO. I did not like that as a,
Okay. So the three groups, there is ND, no diabetes. GADA, I don't want to say that whole name and mess it up, so we're just gonna say people at risk for type 1 diabetes. And then T1D, type 1 diabetes. So what the dots means is each dot is a gut hormone cell. So the purple dots are the cells that make insulin. The other colors are cells that make other hormones. And then bigger dots are just bigger cells. They have more information, but that doesn't really like matter to us right now. So A through L, which is like I think you could see that. Yeah, A through L. Um, basically, the people with no diabetes, their cells have lots of insulin making gut cells. And then for people that are at risk of type 1, they still have a lot of insulin cells, but they don't have as many as someone who does not have any risks for type 1. And then type 1 diabetes, they don't really have a lot of wait, insulin making gut cells left. So basically, what that shows is that insulin producing gut cells start to disappear as type 1 diabetes develops. So then the graphs M through O, what they're showing is graph N is how many gut hormone cells are in total. So there's fewer cells that we could see during type 1 diabetes, and then there's a loss of cells in people that are at risk. So then for graph N, it's the percentage of cells that make insulin. So this is very high in people that don't have type 1 diabetes. And then it's starting to lower when you have the risk of type 1. And then for type 1 diabetes, it's very low. There's a low percentage of cells that make insulin. So graph O is the percentage of cells that don't make insulin. So the percentage of cells that don't make insulin, it goes up when you have type 1 diabetes, meaning your cells just don't make insulin, or don't make as much as they would normally. So the gut isn't losing all of its hormone cells, but it's losing the insulin ones. So what this shows us is that as type 1 diabetes develops, the insulin producing gut cells are lost and they're replaced by cells that make other hormones. So this matters because it shows that type 1 diabetes affects more than just the pancreas because it affects the gut hormone system changes as well. Any questions?
Yeah, why EO? That may not be a question for you, Kira. That
YEO?
yeah, that's probably a question for Sam, but why? Why couldn't they say islets? What was the reason? Why call it an
You know,
EO?
because Sam wrote this paper.
You can say you don't know. That's fine.
Um, I'm trying to think.
Isn't that weird? Like it really
Yeah.
because I read papers differently than you guys do. I read the abstract and skipped straight to figure one and immediately was lost. I had no idea what they were talking about because I don't know what an EO is. So in figure two, it's all about EOs, and I had to go back and read the paper and figure out that all it meant was an endocrine object, right? And then they added a whole bunch of things to it. So ICEO is an insulin containing endocrine object. IDEO is a insulin deficient endocrine object. Endocrine objects, they're just talking about islets, right? Isn't that odd? Now, listening to Kira do her explanation, I came up with a possible reason. If an islet is not producing insulin anymore, but maybe some other type of hormone, as she said that they were being replaced with other hormones, right? Is it still an islet? Maybe that's the reason why, and I'm not an endocrinologist, so I don't know. Does that make sense? If the definition of islet is a group of cells in the pancreas that produce insulin and glucagon and somatostatin, and then all of a sudden you've switched it to some other hormone, is it still an islet? You see what I mean?
But if it's just the beta cells dying off, it would still produce glucagon, right?
Right. But is it still an islet? Words matter. I think it's a good example of maybe I'm being stupid and they were doing a good thing. But I thought I'd throw that out there because all of you have this trouble. Maybe you didn't realize it because you weren't actually trying to process it or read it closely. But for those of you who read it closely, you probably had that problem where you're like, why? What are they? Why are they changing the name? Why are they not just calling it an islet? Right? Because if we go back to the previous figure, Andrew said that the size of the islets were important. But if we're not calling them islets anymore, then right? You see how that could be difficult? Look at the enthusiasm. Oh, come on, people. This is important. Diabetes is important. Diabetes is a disease that will be extinct in 15 years. Okay? We're going to kill all the diabetics. Just kill them all off. Then it's no, I'm kidding. I'm kidding. The
Bye,
disease will be
Sam.
cured. Goodbye, Sam. The disease will be cured, I think, in the next 15 years. Okay, so that was just one train of thought. Here's my second train of thought that I needed for this one and the previous figure. What This is a question for the sophomores, and if they fail, then for Lila. What are the numbers in figure 2M, N, and O? What are the numbers at the top of the figure?
Don't those I'm either gonna look like a genius or a fool, but don't the numbers show like significance?
They do, which is also referred to as.
Okay, now you're really testing my luck here.
Anybody? Anybody remember?
Like the p-value.
That is correct. The p-value. Who said that? Who was that?
It's me, Maddie.
Maddie. Yeah.
Yeah.
Yes, it's the p-value. Um, I'm just gonna say, as somebody who does biostats, that's a kind of strange thing that they did. Because if something is less than 0.05, it's significant, right? So you either it's a digital thing. It's either you are significant or you are not. So then what do these p-values, what are they supposed to tell you if it just means significantly different? What do they, anybody know? Why include a p-value? Lila, you're the only one who might have a chance of answering that.
You're saying Y includes statistical significance?
No. Why include the p-value on the graph? Right? Isn't that odd? Isn't it weird to you that they included the p-values on the graph?
For this particular graph or graphs in general? Because I've seen it.
Graphs in general. I find it odd whenever I see them on graphs. They shouldn't be on graphs.
You don't think so?
Well, it depends on what you're trying to say. What's the purpose of it? So freshmen, we're doing this in the next week. You will be figuring out what the p-values are of things. So this is directly relevant to the experiment we're running. If you can only be significant or not significant, then why show the value? Why not just put an asterisk? Which is generally, right, Lila, that's generally how they do it. It's just like some sort of asterisk. Why include the p-value? Well, the smaller the number, the more confidence you have that it's real. Now, we say 0.05, it's the cutoff for the p-value, and we say, okay, if it's beneath 0.05, it's significant. But if you see a p-value of 0.05 versus 0.005, you have more confidence in the 0.005. I think that's why they did this. But as somebody who has read a lot of these, to me, that also means that maybe there's a little bit of naivete about the statistics that they included this, because did any of you read the methods? Did any of you read, one, who are the subjects, and two, how they ran the stats? Because they actually did something that some of you sophomores did. Actually, Lila, you might have done it too as a freshman, where in the figure legend and in the paper, and then again in the methods, they actually explained which statistical test they ran and why, which usually you don't put in a paper. And to me, that means, oh, this is somebody who's new at statistics, and they want to make sure everybody's on the same page. Like, did you see what test is this? For M, N, and O, what is the test?
I think they did a true school wallets.
Yeah, they did. Do you remember that, sophomores? Because you ran that last year, Kruskal-Wallis. So they ran a Kruskal-Wallis test. However, what are we looking at? It says significance was assessed. Sorry, significant statistical significance. Oh, I'm reading three. Sorry, I'm reading three. Number two. It's at the end of the figure legend, right? Statistical significance. Oh, data are presented as mean plus or minus standard deviation. Come on, sophomores. The data are being shown as means plus or minus standard deviation. What does a Kruskal-Wallis test actually test? Hint, does it test means? Did my thing die? I don't hear anybody. What's going on? That's not what I want. Stop sharing your screen for a second, Maggie. I'm going to show you guys something. Okay. This is
Car,
important. Yes.
car, is the Kruskal-Wallis test the one we tested to see which one was like more superior to the other?
Oh, you're on the right. No, but you're on the right track. You're thinking about it correctly.
Okay.
Yeah, you're thinking about it right. Okay. You guys see this all right? Remember this, sophomores and Lila? This is a really important freshman. You'll see this next week. This is how we choose this is the most basic way to do it. My brother laughs when he sees this. He's a biostatistician. For most of my career, my scientific experiments were so simple, I could use this thing from AP Bio to actually determine what tests to run. And the great thing about this is that it actually goes and gives you like not just which test to run, but also what graph to use. They ran a Kruskal-Wallis and they gave you a bar graph. Do you see the problem? Bar graphs are for means. Means are used in parametric tests if the data are normal. They used a bar graph to show means for Kruskal-Wallis. What were they supposed to run? Box and whiskers. Now, stop share. Now, how many of you picked up on that? And what does that tell you about them? Is that appropriate? Come on, people. Shoot from the hip here.
Didn't you say
If
that biologists were bad with stats?
I think this is an example of biologists, good scientists. I mean, obviously, they have a paper in a really good journal, right? But this is why AI is going, like Nature actually has an AI bot going through its archives to retest all the stats that it ever published. And people have to keep withdrawing their papers because they analyzed it incorrectly. In this case, I don't think it'll make that big a difference, but that really wasn't what they should have shown. They shouldn't have used a mean to show a test for median, you know? And so I really like this paper, not because it's perfect or it's really terrible, but because it does a lot of stuff that you guys will get wrong kind of right in the beginning of your careers. And yet it's still in a great journal, right? So, and if I went and called up the corresponding author, that corresponding author could be like, "No, you're an idiot. I teach statistics here at the University of Florida," because that's where they're from. Not Florida State, sorry. Yeah. But
That's why
University
the
of
stats
Florida.
are wrong. Sorry.
That's probably why the stat. No, and he might be like, "You're wrong because," and then have reasons, and then I'd be like, "Oh, all right," because there was other stuff going on that would make it correct. But on the face of it, it doesn't look like they actually ran the stats right, which is also why Maggie and Sam, why I said, "Where's figure four?" Because they make the same mistake in figure four. Yeah. I don't think it's critical. I don't think that it ruins the paper. I don't think that I don't think it means that had they run it correctly that they would get a different result. I think they end up with the same result. But I'll when we get to the end of everything, I'll maybe point it out maybe in overtime. Okay? So maybe you don't think any of that is interesting, especially if you're a freshman. You're like, none of this means anything to me. But if you're a sophomore, this is an example of what I would talk about in the videos that I did for all your lab reports last year, right? Like, how did you run the stats? Why did you talk about it the way you did? Are you sure you're actually being precise? Because that's important. You don't want your paper withdrawn. I'm apparently losing my voice. All right, go ahead. Sorry.
Okay, Kira, were you all set?
Yeah, I don't really have anything else to say for it.
Okay. Um, figure 3. Does anyone have any? Don't want to take a shot at it? Okay, go ahead, Lily.
Okay, so in this figure 3 and image A, you can see the groups that are being shown are non-diabetic, the GATA group, and then the type 1 diabetics. You can see for the non-diabetics that the insulin positive and GCG negative is a lot more abundant than the insulin positive and GCG negative for the type 1 diabetics. And then similarly, the insulin positive and GCG positive is a lot more abundant for non diabetics than it is for type 1 diabetics. And then the opposite thing, or the opposite pattern is true for the insulin negative and GCG positive. It's a lot less common for non diabetics than it is for type 1 diabetics. And then for the GATA group, they have much more insulin positive and GCG negative than either of the other groups. So, yeah.
Okay. Does anyone have anything to add on to that? Okay. Moving on. We
Well,
have
what was what's the takeaway?
Lily.
Um, that non diabetics have a lot more of insulin positive and GCG negative and insulin positive and GCG positive than type 1 diabetics.
A
And
lot
the
more
opposite
of
is true
them.
or it's like more abundant.
Okay, that's one thing. So spatially, sure. What else?
For the type 1 diabetics, despite having like a smaller abundance of the insulin positive and GCG positive, theirs are a lot larger.
There you go. Yes. That's it. Good. Good.
Is that all?
Yes. Yeah. You
Uh,
should be able to boil these things down into really simple statements, right? Because otherwise it would never get into a journal like this if it was too convoluted and confusing. That was good, Lily.
all right. Dominic. Okay, so Dominic said, I don't know if you already answered this question, but what do the numbers mean next to the images?
Hang on, I have this written down somewhere, I think.
What numbers? Which ones?
In the little yellow boxes. It said it in the description
Oh,
underneath the figure, and I now can't remember what they were.
the
Has to do
numbers
with
in the
the
yellow
density.
boxes indicate the percentage of EO in each size category.
Sorry, I heard someone like call out. Uh,
That
who
was
was that?
Dylan, right?
Was that
No,
Dylan?
I was just saying, I was asking if it had something to do with the density.
Indirectly, yes. Numbers in yellow boxes indicate the percentage of EO in each size category. Because these are categories on the left, right? You got CC, whatever that is, small, medium, and large. And so the large ones, for example, are way higher percentage of those EOs in the type 1 diabetes. Right? That sound right, Lily?
Yep.
Okay.
All right.
Next.
Moving on to figure 4. Does anyone want to give this one a shot? Okay, if not, I know that you mentioned that there is, again, like something wrong with the graphs chart. So if you want to jump in and kind of explain that further.
Wow, you just cold called me. Wow.
Oh, yeah.
So this is a statistical analysis. And if you read the figure legend, it says it says that statistical significance in each EO size category was assessed using a one-way ANOVA with a Bonferroni post hoc test when model assumptions were met. And if you guys remember, sophomores and Lila, that in order to run a parametric test, you have to meet two criteria, two rules, at least two rules, right? One, the data have to be normal, and two, the variances of the data set, how wide the bell curve is, can't be significantly different from each other in order to run the ANOVA. So some of these are ANOVAs, and analysis of variances, ANOVA. ANOVAs are a test of means, so what they plotted, which are means, is appropriate. But when they didn't meet the assumptions, when that failed, which happened last year to us, they have to run a Kruskal-Wallis test, which is fine, except that you can't use the means, which is the mistake they made. But I can't really tell you which one's which because it doesn't say which one was the ANOVA and which one's the Kruskal-Wallis. Yeah, doesn't say. So you just kind of have to go with it anyway. But look at how giant in like C up at the top for density. Look how giant the error bars are. And the asterisks, notice there are asterisks that are like one asterisk or two or three. That's another way of telling us just how confident they are. So if it has one asterisk, the p-value is less than 0.05. Yes, they are significantly different. If it's two, that usually means that it is less than 0.01. And then if it's three, it's usually less than 0.001. And then if it's four, it's like quadruple zero. So it means you have really, really high confidence in it usually for a four. But then they have stacked asterisks like Maggie just circled. Not really sure what that means. Because if you remember, sophomores, that I think we used letters last year to show the differences between things, right? Remember, you had to, if you shared a letter, they weren't significantly different. I think that's what they're trying to show here. I can't tell. I'm not sure.
You mean like the amount of like stock?
What's purple asterisks versus black asterisks versus, well, there should be green, right? But there's not. So I don't really know what that's supposed to be showing. CC is cell clusters. S is small, M is medium, L is large. Ah, here it is. Gray asterisks show differences between the no disease, so the control group, and the diabetes group. And the purple asterisks show differences between the early stage diabetes and then the type 1, the actual diagnosed type 1 diabetes. So what you're looking for is, okay, what's actually different from them? And if there's a difference, then you can say using this method of 3D imaging with light sheet microscopy, you can say, oh, well, that's a good test to see whether they're likely to get it, diabetes. Does that make sense? So like, Sam, have you ever had a biopsy of your pancreas?
No.
Okay. Because remember earlier I said, did anybody read the methods and see who the study's on? Did you guys see that? These are donor pancreases from humans, meaning these people are dead. So you had 8 people who did not have diabetes, who died, and they took their pancreases, and 5 people who got diagnosed with type 1 diabetes. And I don't know what the middle group was. But what they're trying to say is,
Is that what it was at the bottom with like all the contributions?
no, 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 no. That's just if you look at the initials, you'll see those are the authors.
Okay. Because I
Yeah,
saw
they'll
like there
say
was like a list of a bunch of people and I was like, did they just
no, no. But cool fact: if you go to medical school and you need to do gross anatomy, when you walk into the cadaver room, there will be a list of the people's names on the side, which you might be like, wait, that's a breach of privacy. Why would they need to know? Why would they need to know that, who the names are? Which, I mean, that's really interesting. HIPAA and stuff. Like, don't you need to protect their identities? Well, first of all, they're dead. Secondly, they've donated their bodies to science. So you're kind of, you can go do whatever you want. But thirdly, you do not want to be that person who walks into the cadaver room, takes off the shroud, and goes, Grandma. So that's why they do it. They don't want you to actually have like somebody in there. They want you to be prepared. When I was at Vanderbilt, I used to help them flip the bodies, but the cadavers are very heavy. So when they needed that, they go in and flip the bodies with them. Anyway, I think that's kind of the gist of this one.
Okay. Before I move on to the next slide, I just want to say thank you, Lily, Kira, Andrew. Okay. So what? You're looking at me like I'm crazy.
I just did one. What the heck? What am I?
Yeah, but you know what you're talking about, so.
Oh, okay.
All right. So, the
I don't,
implication.
Maggie.
No, you do, Kira. You know a lot. Um, so the implication, so this matters because it could help scientists predict the progression and possibly also the prevention of type 1 diabetes. Sam, do you want to do the scientific take home?
No, wait, pause. This is why I asked about biopsies. Because these people are dead. Can you run this test in you, Sam? Can they test you with this test if you're not dead?
Um,
I would think with a biopsy you could, right?
like a biopsy of like an islet.
Of your pancreas. Okay. Having had a liver biopsy, I can actually describe this. It's a needle this thick. Okay. They jam it into the organ it has a little claw on it. It grabs on and pulls off a piece, and then they take it out. Now, most of the time you're asleep for this. I was not. It was the worst pain I've ever felt in my life. If you ever wondered, what's it feel like to get stabbed in the abdomen? I now know what that feels like. But yeah, so I think that you could use this as a real kind of predictor later on. Okay, good.
So the scientific take home of this paper is that the size and composition of the islets can determine the order of the cell death.
And so we listed our new favorite word as the islets because the amount of times that we mispronounced this word while practicing was extreme. Um, and then, does this paper belong in Nature Communications? We both said yes because we think that
Yes.
it's very helpful. And then the journal impact factor was 15.7. And I think that is on a 2-year
That is, yeah.
basis. Yeah.
Yeah. It's bigger
Yeah.
for a 5-year.
Yeah, 5-year, I think it was like 17 point something.
Yeah, that's really high. And so, because it's really high, what did this, like, really kind of going back to what Dylan and I were discussing in the beginning, was like, what is, what does this paper give you that didn't exist before that qualifies them to have a paper in that good a journal? And I think it's simply light sheet microscopy. I've got this toy, and if you use this toy to look at islets in 3 dimensions, it actually could be a useful tool that we don't have or that we didn't have to determine whether or not you're going to get diabetes by looking at these factors, density and size, right? But
You
cool.
want that to be on like overtime because it's
Mm-hmm.
it's
Yep.
9:05. So
Yep.
thank you guys. And yeah, you guys are free to go now, but there's overtime. So
Good job,
go get
guys.
stuff.
That was really good. Good job. Okay.